We will start with the webinar in a few more minutes. We are now live on YouTube. We'll wait for maybe a few more participants to join and then start.
Yeah, okay.
Okay, I can see participants have started joining. Hello, everyone. Welcome to the webinar. We will be starting in a few more minutes, 1 to 2 more minutes, and then we will start with the session. And for those of you who are just joining right now, please feel free to introduce yourself in the chat box. Do let us know where you're from and what is your educational background. Are you a student or like a working professional? Please do let us know in the chat box. Thank you. Okay, so without further delay, I'll slowly start with the introduction, and meanwhile, the other participants can join. Let me start with the recording first. Okay, so I hope the participants will join in the meantime, and I'll just start with a brief introduction to what this webinar is about and what you can expect from this webinar, and also some exciting scholarship opportunities and some research opportunities that the participants can explore at the end of the webinar. So I hope everyone will stay till the end to hear about the exciting announcement. And with that, without further delay, I'll just start with a brief introduction. So, hello everyone, a very warm greeting to one and all, and I would like to welcome you all for today's webinar on mapping molecular signatures in liver cancer, which is a student project by Pavani Annambhotla. So, she is an alumna of the OmicsLogic Biomedical Data Science Research Program who worked on the research project titled Gene Expression Signatures in Hepatobiliary Carcinomas. And in this session, Pavani will walk us through her data-driven exploration of gene expression patterns in hepatobiliary carcinomas. She'll also share how this research experience and mentorship helped her strengthen her bioinformatics skills and confidence to pursue research opportunities. And the session will also, as I mentioned, will introduce some exciting research opportunities that if you're someone who's interested to work on some bioinformatics research project and would like to work under the guidance of a mentor, then we will be announcing some opportunities that you can be a part of, just like Pavani, and work on research project, and also some scholarship opportunities that participants that is exclusive for the webinar attendees can avail. Okay. So please stay tuned till the end of the webinar. So I would like to take a brief moment to introduce you to the team behind the webinar. So we are a global bioinformatics and data science company with roots in India and the US. And over the years, we have had the privilege to collaborate with leading universities, research institutions, and industry partners worldwide. And our mission is to bridge the gap between data-driven research and skill development. To achieve this, we work with regional and international experts who help us tailor our program to the specific needs of students, educators, and researchers across the globe. Now, with the growing demand for skilled bioinformaticians and data scientists, we have developed a range of training and research offerings, including online courses, specialized workshops, internship and fellowship, research services, and even a degree program that is dedicated to bioinformatics and omics technologies. Now, here's a quick glimpse into what a student learning journey looks like. So, most of the participants who are interested to work on bioinformatics research projects, they are complete beginners, mostly from life science background. You also have working professionals who are trained in wet lab and would like to now transition to dry lab aspect. So here's a quick roadmap that you can look into how this works. So it begins with training in hands-on courses and programs that is suitable for all levels, so even if you're a beginner or even if you're an advanced learner. And for those who are looking to go beyond, we also have the research program that Pavani was also part of, and you can work on real bioinformatics research project under the guidance of a mentor. I'll be explaining more about the research program in my upcoming slides, as most of the participants might be curious to learn more about the research opportunities. Now, we also offer courses and example projects that is learning based on a diverse range of topics from precision medicine, oncology, infectious diseases, metabolic disorders to even agri-bioinformatics. And all of these courses are enriched with engaging content, including visuals, infographics, and video lectures that simplify the complex bioinformatics concept for better understanding. Now, we also heavily focus on experiential learning through short tutorial, practical demonstrations, and one of the most favorite part of the participant is to work on coding exercises using Google Colab Notebook as well as through inbuilt apps. Now, while our courses are fully automated and lesson-based, we also understand the value of mentor-guided instruction. So that is why we also offer workshops and training programs that combine mentor-led sessions with hands-on walkthroughs to reinforce several of the concepts. So this also includes a combination of expert video lectures, interactive tutorials, and step-by-step lessons. And all of these are curated to ensure that the learners get both the best of both, that is theoretical knowledge as well as the practical application. Now, this is the research program that I was talking about in the beginning of my presentation. So some of you might already be familiar about the research program and might already be in touch with our team who's interested to work on some of the projects. But for those participants who are joining with us for the first time, I'll just give you a brief introduction about the program and some of the outcomes that we have seen. So the mentor-guided research program provides an unparalleled opportunity for participants who are interested in working on research. And these could be students, researchers, citizen scientists across the globe to work independently on a bioinformatics research project. And with mentor support, this program goes beyond the traditional learning, allowing you to delve into real-world bioinformatics research using cutting-edge omics data. So you could choose to specialize in a specific omics field, and you will be initially trained. I'll be showing that in a roadmap in my upcoming slide. So you can choose to specialize in a specific omics field like genomics, metagenomics, transcriptomics, cheminformatics, metabolomics, and then further you can also choose to choose a specific topic for your project, including it could be infectious diseases, metabolic disorders, cancer research, which is also one of the most popular amongst the student. Neuroscience is also another interesting topic that we have seen from students, and immunology, astrobiology, and so on. So how does a research program works? So here on the screen, you can see a program roadmap that clearly outlines the student's journey throughout the program. So as you can see, it begins with students undergoing a comprehensive training in their chosen field of omics. So like I mentioned, this could be genomics, transcriptomics, metagenomics, and so on. And the objective here is to help the participants build a strong foundation in key skills such as data processing, analysis, and visualization. And in the next stage, students will work on identifying an impactful research topic through regular mentor meetings and in-depth literature review. And now in the third step, students will work on data collection, analysis, and interpretation. And this is also the stage where students will start looking into data sets to identify trends, patterns, and correlations that will be further critical for your project work. And finally, the program culminates in students working on continuing the report detailing all the analysis and interpretation they have carried out, preparing presentation, and submitting their final work to the mentor to receive expert feedback. So you can see a glimpse into all of this during the presentation by Pavani. She has also went through the same program outline. And I would also like to take a quick moment to showcase some of the examples on how students from previous batches have leveraged these training and research opportunities to achieve their academic and professional goals. So here on the screen, you can see a cohort of students from System Medicine graduate program at Georgetown University who completed their training in Python and further worked on various machine learning projects to address biomedical research challenges. Now, students also have went on to present their projects in international research symposium and poster presentation competitions to win exciting cash prizes. This is an example of a student, a high school student, who worked on an Alzheimer's project and who later on went on to win the Potemkin Prize, and is currently enrolled in Harvard University. So there are different participants from different backgrounds who enroll, including high school students as well. Now, students also join the program to get research publication done, and in my few slides, I'll just show some of the areas where some preprints and publications have been carried out in the hope that that might inspire you to work on similar projects. And that gives them this extra competitive edge over other students while applying for higher studies or in the job market. And one of the unique highlight is that faculties and researchers have also collaborated to carry out different publication works. Now, we also get a lot of interest from participants who want to combine science and business. So that is why we also offer an entrepreneurship and business innovation track. So here on the screen, you can see a team of industrial researchers from Nigeria who completed their initial metagenomics training, and then later they went on to develop a microbiome test kit to improve the fertility of women in Nigeria. So all of these projects are also available on our portal. I'll take you to the portal in just a while in case you're interested to explore and learn more about what they have done, how they have done, and what are the various outputs they have got. Now, here's a quick glimpse into the different preprints and publications that have been done in different areas like stem cell, space medicine, cancer research, again, one of the most popular area, neuroimmunology, vaccine efficacy, antimicrobial resistance and global health, COVID-19, reproductive health, structural biology and cheminformatics, metabolic disorders, and this preprint has also been carried out by a high school student who initially just completed the program. And these are some of the feedback that we have received throughout the program and our training over the years. So now I'll just quickly stop sharing and take you to the platform in case anyone is interested to check out any of these student research projects that have been carried out. And let me share my screen once again. So this is the OmicsLogic platform. Now, apart from the research projects, there are also different resources, that is training resources that you can explore depending on your interest. I'm sharing the link in the chat box. A very simple process to create an account. It is free to create an account on the platform. Just head over to the link that I've shared in the chat box. You can click on login and you can click on create an account in case you don't have an account yet. And here you can enter your name, email ID, password, and you can create an account. Or if you already have an existing social media account like Google, Facebook, Apple account, GitHub account, or even Twitter, then you can simply click on it and create an account as well. Okay. And if you already have an account, simply enter your existing email ID, password, and you can log in. And in case you have forgotten your password to the account, simply click Forgot Password after entering your email ID, and you will get an option in your inbox to reset the account. Okay, so with that, I'll just log into my existing account just to show you some of these training opportunities that you can explore as well. And for those interested to take it up a bit further and work on research projects, you could do the same. Okay, so once you log in, the very first interface that you see is the course library section. And here you can see different areas that you can choose to complete your training in. So for example, bioinformatics is suitable for those participants who are coming from life science background, wet lab background, who are completely new and would like to learn the very basics of bioinformatics and just to test the waters if you're interested to work on it. And for those who are already well equipped, who are already in the field and would like to specialize in a specific omics field, they could choose to check out any of these options as well. And there could be genomics, transcriptomics. Now, if you're someone who is mostly into technology and coding, then you can check out machine learning, Python. We also have courses on R coding for omics data analysis, metagenomics, cheminformatics, structural biology, single cell RNA-seq data analysis, epigenomics, Linux, and GitHub as well. So all you have to do is simply click on the Explore button and it will redirect you to a specific set of courses that you can check out as well. Okay, and you can simply click the course open, and here you will be able to see a comprehensive collection of lessons that you can explore. And these contain textual content, there are infographics, visuals, and there are also at the end of each of the lessons, there are some assignments and coding exercises that you can check out as well. Okay, so this is just a quick introduction that I wanted to give you. And whenever, for example, transcriptomics is one of the most popular amongst the students, so I'll just open that coursework as well in case you're interested to explore. It is course 5. And here also you can check out some of the lessons and understand what all are the topics that is covered in the curriculum. And wherever there are hands-on exercises that is involved, there are detailed instructions about how to perform different analysis. Initially, you're introduced to the data set, where it is taken from, the research context behind it. And then we also walk you through the different analysis codes, the explanations for that, different outputs that you get, and the interpretation of the biological interpretation of these outputs. So this is just a quick introduction that we wanted to give you about some of the training opportunities. And this is also something that Pavani had initially completed in her training phase. And now I'll just pass over the stage to Pavani to briefly introduce herself and also talk a little bit about her learning journey from transitioning from biochemistry background to bioinformatics. Over to you, Pavani.
Uh, Good evening, one and all. I'm Pavani Annambhotla, and I graduated from the University of Madras with an MSc in biochemistry. I have an immense interest in various modules like molecular biology, cell biology, microbiology. Like, besides wet lab, I recently transitioned into dry lab. Like, it's not a complete shift from wet lab to dry lab. I thought the combination, the wet and dry lab combination, helped me to gain a number of opportunities. Over the years, I worked in a few wet labs where I developed the knowledge in various molecular biology techniques like PCR, the DNA isolation workflows, and other wet lab techniques. But I felt there are a few questions that I want to answer. For them, I thought a computational workflow is something that would definitely help me to answer my questions related to biomedical research. So I started since an year, I started working on a few tools related to bioinformatics domain. OmicsLogic helped me a lot to learn these tools and skills. I didn't master in everything, but I'm exactly in that beginner to intermediate level where I started learning various tools from various platforms. So in the further slides, I'm going to explain you how I did my project. So this journey helped me a lot. So I thought this can even help you guys to pursue your future endeavors. So yeah, without any delay, let's go into our slides. So my project is gene expression signatures in hepatobiliary carcinomas. My transition from biochemistry to bioinformatics, it's a very natural one. While biochemistry, where it gave me a deep understanding of basic knowledge in biology like proteins, enzyme estimations, other cellular pathways, and various other mechanisms, where these days modern biology is generating massive data sets. So exclusively in various domains like genomics, transcriptomics, proteomics, etc. So I realized that to truly understand any disease mechanism, I thought I should get into these skills no, besides the lab bench. So bioinformatics is a domain, it is a powerful domain, I think. So it helped me to learn more about what is happening inside a cell. Um, so being able to combine wet lab knowledge with computational analysis, it turned into a huge advantage for me. I understood that if I wanted to contribute something meaningfully to any kind of research, uh, say precision medicine or drug discovery, whatever it might be, I needed to build strong computational skills. So the transition, it's more like expanding my ability to answer bigger biological questions using data. So my motivation to move from wet lab to dry lab came from any number of research problems I wanted to explore. Wet lab experiments are powerful though, but they were time consuming, expensive, and sometimes limited in scope or availability of the resources. When I started working with real world biological data sets, I realized that I could analyze thousands of genes, proteins, or mutations related to a single experiment using computational workflows. For example, Galaxy can analyze a large number of data sets at a time. So this efficacy and depth made me computational biology extremely appealing to me. And this motivation pushed me to pursue domains like transcriptomics, genomics, machine learning, computational methods, which eventually led me to do projects like hepatobiliary carcinoma analysis and other few computational biology related projects. So I planned it step by step. Like I first started learning basic programming languages like Python and statistics. Of course, in whatever domain it might be, statistics plays a key role. Like at a glimpse, it can give us an overview of the data set. So and Linux, PowerShell scripting, even though they were a bit complicated, but having a minimum knowledge in those languages is most essential for computational workflows. Then I began exploring real world data sets from platforms like NCBI, GEO, ENA, and I started working with public data sets for a mini kind of projects, which helped me to build confidence and understand how biological data is structured. And next, I enrolled in multiple structured programs like transcriptomics training and how to design a research project uh, from OmicsLogic platform. And I started doing mini projects in domains like AML for biology, gene expression analysis, and I learned tools like DSEC2, Galaxy, T-Bioinfo platform, Veka, Kimi2, etc. So practicing these regularly helped me to do projects in an easy way, besides purely wet lab student. So one of the reasons I was able to transition successfully because I constantly upskilled myself through various certifications and trainings. I completed multiple programs, including immunoinformatics, the gene editing, RNA related domains, NGS workflows, on the computational side. So each certification gave me a specific skill uh, for handling omics datasets, applying machine learning algorithms, and to build a kind of CADD model, a computational aided drug design model. So these courses created a strong foundation for me, just like the wet lab, and especially in domains like AI-driven biology and cancer bioinformatics, etc. One of the turning points for me was getting trained under Professor Graham Ball, associated with Nottingham Trent University. So he ha he trained everyone in how machine learning algorithms can be applied to real world biological data sets. That program taught me from the scratch, like from data pre-processing, model building, classification techniques, and result interpretation. So it was the first time for me. I realized how powerful AI can be when it is applied to genomics and healthcare. As a part of that course, I worked on a mini project using Veka tool, which is developed by University of Waikato, where I compared multiple classification models on biological data sets. I used techniques like decision trees, random forests, logistic regressions, naive Bayes to understand their performance. That mini project helped me to build confidence in ML and pushed me towards to do larger projects like this. So I have always been fascinated by cancer biology, oncology, because it's a disease that is affecting since eras and eras, affecting millions of people. It remains extremely complex. Cancer is not just an uncontrolled cell growth. It's a combination of a number of mutations, microenvironment changes, immune evasions, metabolic rewirings. So much will happen in our body if we are affected with that. So understanding the complexity of the disease is what motivated me to explore cancer genomics through different data sets and computational tools. So I chose liver cancer specifically because it combines two areas I'm very passionate about, like immunology and fibrosis. Hepatobiliary carcinoma grows in a background of chronic inflammation. Hepatic stellate activation occurs and ECM remodeling occurs. And I thought it is a perfect model to study both tumor biology and surrounding a microenvironment too. That's what made me to pursue HBC project. And I excitedly done with that project. Before that, I started taking the transcriptomics training for that. I took a 2-month training program from OmicsLogic platform only, where I learned everything from raw data pre-processing to normalization, PCA, DSEC2, and other pathway analysis. It gave me confidence to handle real-world data sets. The training made me a big difference because when I started HPC project, I already knew how to interpret gene signatures, pathways, PCA clusters, and other enrichment results. So instead of struggling with various tools, I could focus on biology, which ultimately made the project stronger and more meaningful. In the HBC project, I performed complete transcriptomics workflow. This included data cleaning, normalization, PCA analysis, and identifying differentially expressed genes using dissect tool. I generated volcano plots, heat maps, clustering visualizations to understand how tumor and normal samples differ. After identifying key genes, I performed gene ontology, KEGG pathways, reactome, and other various pathways using iLinks platform. I took dataset from NCBI and where I have both tumor and control liver tissues. I have done this project from the scratch, like as I said before, from raw data cleaning to data visualization. So this helped me to identify pathways involving immune response, ECM remodeling, collagen formation. I got to know so many points after doing this project. This entire workflow provided a detailed biological understanding of hepatobiliary carcinoma. So PCA, it is a principal component analysis. It is a statistical method that reduces complex gene expression data into a few components that explain the most variance. To put it in simple, it helps us to visualize whether tumor samples behave differently from normal samples or not, like at the gene expression level. To perform PCA, we first need to normalize the RNA sequential data, then apply PCA algorithm that is provided by Bioinfo platform, which that computes the variance and projects the data into 2D or 3D coordinate system. In my project, PCA clearly separated tumor and non-tumor samples, proving that data sets were um, biologically meaningful and suitable for further analysis. Differentially expressed genes. These are the genes that show significant differences in expression between two conditions like tumor versus non-tumor. DSEC2 tool identifies differentially expressed genes based on fold chain and adjusted p-value. So it helps us to find which genes are upregulated and downregulated. These genes reveal important biological processes. For instance, in liver cancer, the upregulated genes were related to inflammation, cytokine signaling, fibrosis, ECM remodeling, etc., whereas downregulated genes were related to very normal liver metabolism. DEGs are something that essentially tell us like what's going wrong inside our business system. That's what I found using DCP. And on this side, top differentially expressed genes are something that like they give us a question how they connect to cancer biology, specifically hepatobiliary carcinoma. These genes stood out consistently in the analysis due to their strong dysregulation patterns. Each gene plays a specific role in tumor biology, ranging from cell signaling, metabolism to stress response pathways. Understanding these genes and with understanding these genes helped us to helped me to piece together various molecular landscapes together related to the disease and how different biological processes come together to support tumor initiation and progression. For an instance, DKK1. This is a gene which is a key regulator of WNT signaling and strongly associated with fibrosis and other cancer invasions. Similarly, the two other genes, GRI4 and KCNU, they reveal interesting connections to neurotransmission, apoptosis, and metabolic reprogramming, and how cancer cells can hijack normal cellular functions like throughout the body. Meanwhile, other genes like PDI2, MEP1A, they are connected to stress, ER stress, and protein processing mechanisms. Altogether, these genes represent potential biomarkers, um, uh, like helped me to find out therapeutic targets that may support more precise diagnosis and treatment strategies for HPC. Pathway analysis, KEGG pathway. Uh, I used iLink's platform where I got to do all these things. So the enriched pathways identified from the KEGG analysis using Enricher. These pathways reveal how upregulated and downregulated genes converge into biologically meaningful processes. Interestingly, many of the enriched pathways overlap between metabolic reprogramming, neuronal signaling, immune dysregulation, lot of things, all of which play important role in cancer progression. This gave me a clear picture of how hepatobiliary carcinoma operates at system level. It's very dangerous. And KEGG pathway. This bar graph visually represented the enrichment scores of top KEGG pathways that are associated with differentially expressed genes. It allows us to compare the relative significance of each pathway, highlighting which ones are most strongly impacted in HPs. For an instance, nicotine addiction. See, nicotine addiction and neurodegeneration pathways show particularly high enrichment, suggesting strong involvement of neurotransmitter-related mechanisms. And gene ontology. Gene ontology enrichment results across three major categories: biological process, molecular function, and cellular component. These classifications help to break down complex gene expression patterns into more understandable biological functions. By examining GO terms, we gain a deeper understanding of how cancer cells reorganize their biological process to sustain growth and survival. So key findings derived from the gene ontology analysis brought together the most relevant biological process, molecular functions, and other cellular components. It helped to consolidate what the analysis reveals about tumor behavior, particularly when it comes to strong involvement of WNT signaling, metabolic shifts, neuroimmune crosstalks, whatever they are. They also showed central themes like altered fatty acid metabolism, which reflects the metabolic reprogramming. Because neurotransmitter receptor activity linked to the signaling changes to tumor dynamics and lumen enrichment highlights the protein folding stress, which is a survival mechanism for tumor cells. Together, these findings map a multi-layer picture of hepatobiliary carcinoma shaped by fibrosis, metabolism, stress adaptation. The scores of the top terms across BP, MF, and CC categories. This provided an intuitive way to see which biological themes are most prominent. The highest enriched terms, WNT signaling regulation, synapse organization, and the receptor activity supports the earlier functional insight. So by quantifying enrichment scores, these visualizations strengthen the confidence in earlier gene ontology-based conclusions, which reinforce the idea that tumor progression is not something that is driven by single pathway, but an integrated network of biological processes. Gene mania, it's a kind of game changer where it illustrates the connections between the DE genes and the interacting partners of the disease. This network approach is something powerful because it reveals co-expression clusters and functional modules that may not be obvious from individual gene analysis. Because it demonstrates visually like how genes are interconnected to each of them through shared pathways. Identifying these clusters is valuable because they may represent regulatory hubs and potential therapeutic intervention points. In summary, my project highlights how transcriptomics can reveal meaningful changes in the gene expression and uncover biological pathways driving hepatobiliary carcinoma. By combining the differentially expressed gene analysis, enrichment studies, and other network modeling sort of things, I was able to identify key signaling pathways and potential biomarkers. The integrative approach shows the power of bioinformatics in understanding complex disease pathways and guiding future research directions. To summarize my journey from transitioning from biochemistry to bioinformatics was one of the best decisions I made these days. Because the world is moving towards data-driven biology. Data science is nowadays, it's a very crucial subject for everyone in whatever sector it might be. The students today most equip themselves with both wet lab and computational skills. Bioinformatics is not, it's not difficult. It just requires patience, practice, curiosity, and consistent learning. For students who want to start, my advice is to start simply, begin with one short data set, learn foundational tools like PCA, DSEP2, understand the biology behind your results. Like try to align both wet lab and dry lab. Keep improving your computational skills. Programming languages are something, they were very important. Every project you do will definitely make you stronger. Start with mini projects, like using a single tool, a single data set, just like that. Like the goal is to not to master everything in a single day or one night, but to slowly become confident with whatever the data we are pursuing with the tools, the scientific interpretation, the first, it entirely depends upon our perception. So my conclusion is to you guys to develop more interest in bioinformatics because it helped me to ace a few opportunities, internships, etc. And right now, I started training, getting trained in immunoinformatics module. I'm working on a project called monkeypox related project. I mean, monkeypox virus research project, where I'm analyzing genomic features, immune signatures associated with infections, kind of infection biology. This project is going to give me deeper insights into viral genomics, host pathogen interactions. I'm also working on multiple research manuscripts, including projects on SARS-CoV analysis, variant modeling, cancer gene signatures, etc. So the upcoming projects reflect my long-term goal of contributing to infectious disease genomics, cancer research, and AML-driven biology. I believe each project is prepping me to become stronger computational biologist, and I'm excited to continue and exploring the new research areas. So for this, I recently began my training in immunoinformatics. The goal is to understand how these computational tools can be used to predict various immune responses, identify vaccine targets. Like we can build computational aided drug design. We can build a vaccine using epitopes. We can analyze epitope receptor interactions using this immunoinformatics knowledge. There are different platforms like IEDV, IMport, where we can learn about various data sets. We can approach in silico pathways. We can work with epitope prediction tools that uh, that they can focus on antigenicity, allergenicity, toxicity, like antigen antibody reactions, MHC binding affinity. There is a lot more. Like we can explore a lot. To deepen my understanding, I have been actively reviewing various research papers to see how these tools are applied in real-world immunology and vaccine design studies. This step-by-step workflow from selecting like target proteins, predicting epitopes, constructing vaccine models, and validating them computationally is what my roadmap kind of roadmap. Yeah, going through the multiple published studies exposed me to best practices, face common challenges, and like how different tools can be used to in a complex way, comparing each other tool, the data sets, everything, whatever. This training helped me to build a strong foundation to eventually carry out my immunoinformatics project with more confidence and accuracy. And finally, to conclude, I worked under Dr. Ritika Patel, ma'am. Um, Her guidance, mentorship, she's very patient. She supported me all the time throughout my project, from the scratch. I literally don't know the before year about anything related to bioinformatics. She is the one helped me a lot. Uh, and today I'm in such a position that, to sit in front of you and speak about my project. She helped me a lot. And of course, OmicsLogic, definitely need to thank you a lot. It's such a very good platform. It gave me a number of opportunities and providing different resources, different kind of subscriptions, different modules. Day by day, they were focus. They were running along with the global opportunities, global knowledge subjects, whatever. So yeah, special thanks to all my colleagues, collaborators, everyone. Thank you.
Thank you so much, Pavani, and thank you so much for the wonderful presentation as well. And I think a few participants had joined the session a bit late, and I'm sure the participants who are listening to the presentation so far might have some questions for the speaker as well. So, in case you have any doubts, please feel free to start posting them in the chat box. Meanwhile, I'll just quickly recap a little bit about the program once again for those of you who joined a bit late, and also talk about some of the ongoing project opportunities. If anyone is interested to join, I can take a brief moment to go through that as well, and also announce some exciting scholarship opportunities that you can check out as well. So, for those participants who joined late, Pavani presented her research project that she worked on under the OmicsLogic Biomedical Data Science Research Program that provides you get with an opportunity to work under the guidance of a mentor on a specific omics bioinformatics project. You can choose to specialize in a specific omics field like genomics or metagenomics, and you can also choose the topic that you're interested to work on, like cancer research, like the one that Pavani did, or even infectious disease, agri-bioinformatics, neuroscience, and so on as well. And we also understand a lot of the students have been reaching out to us with the end semester project, and also they want to carry out the project as a part of their dissertation and as well as part of internship. So we have some exciting opportunities that you can check out, and you can do it as independent project. Also, if you have some peers that you want to work with, then you can also offer the group research project. Now, here's a quick glimpse into some of the projects that you can choose to enroll in. So you may take a note in your notebook in case you see a topic of your choice, but otherwise at the end of the slide, I'll also launch a poll and you can choose the topic that you can that you're interested to work on. So first of all, there is a between 45 to 60 day bulk RNA-seq project that you can choose to enroll in. And here the objective will be to identify gene expression signatures in systemic lupus using bulk RNA-seq data. And another project that we have currently opened is a 60-day single cell data analysis project. And the objective here is to characterize tumor and microenvironment cell types in squamous cell carcinoma using the single cell RNA-seq data. And through all of these projects, you will be guided by the mentor. Initially, there will be training, followed by which there will be mentor sessions that will be scheduled so that you can work alongside the mentor on the project. And another project is a 60-day metagenomics project. So here you will be comparing the gut microbial community composition using 16S RNA-seq data. Basically, it is a human longevity-based project. And we also have another machine learning project. So if you're someone who is interested to mostly focus on technical aspects, then we have a 60 to 90-day project where you'll be developing machine learning models to predict biomarkers from RNA-seq data. And lastly, we have another 45-day genomics project where you will be analyzing somatic mutations and driver variants using MAP files. So if you find any of these topics interesting, you can either get to reach out to us. We'll share our details in the chat box once. So you can just note down any of these contact numbers. You can also drop us a message on WhatsApp. And if you're interested, you can also email us. Our team will just share the contact details in the chat box. And now on the screen, you can see a poll. I'm just launching the poll. Just give me a second. And you can select the topic of your choice that you want to work on. Under the research project, I'll also read out the options once. Okay, so now on your screen, you might be seeing a pop-up that says webinar poll. And I'll just quickly read out the fields so that if you're on your mobile phone, you can just check for the pop-up as well. And if you're on your laptop also. So the first question is to enter your name so that we can know who is filling out the form and we can address you. Just make sure the second field is to email ID. So just make sure that you enter the correct email ID. So the ones that works, sometimes participants tend to enter like wrong email address. So just make sure that you enter the correct one. And the third question is a very important one. Please choose the duration of the project that you want to work on. So it could be 2 months, 3 months, 6 months. It could be maybe like a 45-day project. And you also have participants who work for longer duration, like a 9 months project. So you can select that as well. And in case you don't see any of these topics, just click on others and then you can just save it. And the fourth question is to select the area of the project that you're interested to work on. So we understand a lot of the participants have different research interests. Like I said, like agri-bioinformatics is there, cancer research is there, neuroscience is there. So we have listed some of the common fields that we have seen that the participants are interested in. Once again, I'll just read out these fields. So this includes precision oncology, and another field is infectious diseases like viruses, host pathogen interactions. The third option is drug discovery and structural bioinformatics. And the next option is neurodegenerative and cognitive disorders like Alzheimer's, Parkinson's, and then the option is machine learning based projects. Another one of the most popular emerging topics that we see amongst the students. We can choose to opt for that as well. And then we have the CAR T cell therapy and immuno-oncology, which is mostly immunology-based topics. And like you will be working on like patient response prediction, tumor microenvironment studies, and so on. And then you have the metabolic disorders like diabetes, galactosemia, and so on. And if your topic is not there, if you want to work on a project but it is not there listed, you can also choose the others option. And then in the fifth question, it is optional. Just mention the topic that you're interested to work on. That is not listed in the above topics. You can just mention that in the others option. And then our team can also reach out to you based on the information that you enter here. So I'll just keep the poll open for just few more minutes for other participants to join. I can see some of them are still writing. And meanwhile, I'll start sharing my screen once again. And as promised, I'll just take you to the scholarship form that you can, I'll paste the link in the chat box as well once so that you can access it. And I'll also share my screen once. Okay, thank you so much, Shubhanga. Participants have also started posting their queries, so we'll get to them in just a while. Taya is not able to find the pop-up. No problem. So what you can do is we have shared a link in the chat box, a form, the scholarship form. So you can also choose to submit your interest through the form as well in case you're not able to see the pop-up. Okay, so let me share my screen once and take you to the scholarship form. Okay, so as a webinar attendee, you are eligible to apply for a scholarship while you enroll in the program. So you can apply for this. It is completely optional. And for those who are interested to apply, you can do so. So very basic details, once again, email ID, full name, phone number, country, and company, organization, or universities. And if you are a part of a university or organization that we are associated with, then you will be given further consideration for the scholarship. So just please make sure that you enter your correct company or the university or organization. Also mention your level of education, like are you a high school student, undergraduate, master student, doctorate, postdoctorate, faculty, scientist, or any other options. You can check on others. And also mention, do you have a prior background in the field of research? Yes or no, you could mention that. And the type of research path that you want to opt for. So like I mentioned, some of the students are interested in working on a project independently. You also get a lot of questions from participants who want to do group projects with their friends or their peers. So in that case, you can click on group project. And also, if you selected group project, just make sure to mention the number of students in your group that you want to work with, like 2, 3, 4. Even if it is a large cohort, just mention others and also specify the number. Okay. And also, once again, you can mention the duration of the project that you want to work on. And this is another important field. What is the motivation for undertaking this research project? Is it for as a part of university dissertation work, or is it for publication in scientific journals? Is it for to gain research experience or the other? You can mention that as well. And finally, please write a few lines about why we should consider you for the scholarship. And this field is completely optional, and the last field is also completely optional to upload a CV. So just make sure that when you upload your CV, it is your latest and updated CV and with the correct details so that our team, when they are evaluating, we have the up-to-date information about your background as well. Okay, so this is the scholarship form. This form will remain open till 2nd of December. So we will also email you the link to the scholarship form in case you're not able to access it. And in case you're still not able to get it, you can drop us a message on WhatsApp or on the email ID provided above, and you can share once again with the link to you. Okay. So just as a reminder, once again, the form will remain open till December 2nd. So please take your time to fill out the form and submit your scholarship application. And the poll, however, I'll be closing in just about a few more minutes. Let me take a look. Shall I close the poll or shall I keep it open? Okay, I think few are still submitting. Anyone else who's still submitting? Please take a moment to fill it out. Meanwhile, I'll just start looking into the questions in the chat box. Okay, thank you so much, Ismail, for the wonderful words. And we do conduct such webinars once a month. So please stay tuned. We also have another one coming up in December. So if you're interested, please feel free to join. Another question is from Taya. I need collaboration for the wet lab experiment. I do the genome-wide analysis of gene family in citrus and want a researcher to perform the expression analysis. Yes, we also provide support for transcriptomic gene expression data analysis as well. If you could just drop us an email, or can you share your email ID in the chat box, Taya? And our team can connect with you and understand further about your research requirements, what kind of data you have, and assist you further. There is another question from Shara. Thank you for the insightful session. May I kindly know for participation or attendance certificate? For the webinars, however, we do not provide certifications, but we do offer certificates for coursework, workshops, programs that we conduct. Okay, thank you so much, Thaya, for sharing your email ID. Our team will connect with you on this email ID. If you have a WhatsApp number, you may share that as well. Our team can quickly connect with you on WhatsApp. Okay, Pavani has also shared her LinkedIn profile. So if anyone is interested to connect with her on LinkedIn and improve your network, please feel free to connect. Thank you so much, Pavani. Any other questions about the program? Thank you so much, Thaya. Thank you so much, Zaya. I'll be closing the poll just now, just a second. If anyone else left, do let me know in the chat box. I'm just going to close it. Okay. Thank you, Faiza, for submitting the scholarship form. Our team will review and get back to you soon. Thank you, Gorak. Okay, I'll just close the poll. Just give me a second. Thank you so much, everyone, for joining. Okay. Thank you so much, Arun. Thank you, Noor. Thank you, Ayub. Good to see you once again. Okay. I think if you have still any further questions, once again, please feel free to connect with us. I'm just sharing the contact details once again in the chat box. Please feel free to connect with us on WhatsApp or email ID, and our team can assist with any further questions that you might have about the program. Thank you, Pawani, once again for your wonderful presentation, and thank you to all the participants for your patience and attending. We will catch up again in some other workshop or webinar, and stay tuned. Thank you.
Thank you, everyone. Thank you.
Thank you.